Thank you everyone for joining today. For those who don't know me yet, I'm Alex from the Molecular and Genomics Informatics Core. We also have online today Pat Soteropoulos from the Genomics Center and the team from Nanostring, who are here to talk about their awesome spatial technology for spatial transcriptomics. But before I turn it over to them, just the general housekeeping items. If you have any questions, please drop them into the chat or the Q&A box, and we can monitor that and try to answer them on the fly if possible. If not, we'll ask any at the end. But with that, I'll turn it over to the Nanostring team, and let's hear about the spatial.
So I won't be talking as much. So my name is Matt. I'm the local salesperson based in Philadelphia, and I cover records for all of Nanostring. And then I'll pass it over to Dr. Amy Waba.
Yeah, hi everybody. Thanks for joining us today. And thank you so much for inviting us, Alex and Patricia. So yeah, we're here to talk to you a little bit about GeoMx, our digital spatial profiler at Nanostring. So whether you're familiar with Nanostring or not, I can kind of take you through just an initial look at kind of what Nanostring has been doing and where we feel like GeoMx could be a really beneficial platform. So just to give you a little bit of background on Nanostring, we are headquartered in Seattle. We have about 800 employees, and we have been a company with several platforms since about 2008. So in 2008 is when we launched our nCounter platform. So if you've heard of Nanostring, more than likely that's probably where you've heard of us. This is a bulk gene expression platform, allows you to take extracted RNA or DNA and interrogate your samples that way. And what really made Nanostring special is that our chemistry allows us to be really compatible with a lot of different sample types, including traditionally very difficult sample types like Formalin-Fixed, Paraffin-Embedded, or any other really highly fragmented RNA tissue or sample types. And because they were so tolerant of these different sample types, that's kind of where Nanostring became the gold standard in these difficult sample types. And because of that, we were able to take this chemistry and move into the arena of spatial biology. And so in 2019, we launched the GeoMx Digital Spatial Profiler using a very similar chemistry so that we can now interrogate tissues that are traditionally difficult to interrogate, especially on the RNA side. And then we are now we're about to launch our third platform, which is the CosMx Spatial Molecular Imager, which will then allow us to actually image these tissues and get even higher resolution in the spatial context. To date, we have over 5,000 publications using nCounter GeoMx, and we actually just had our first publication with CosMx. So just a lot of data. We'll be going through some of it today on how you can use these different platforms, but we'll be focusing on the GeoMx. So I mentioned only briefly, but GeoMx is a multi-analyte platform, so it allows you to interrogate in a high-plex manner either RNA or protein, and the chemistry is going to be very similar on both sides. So on the protein side, we're going to bind directly to the protein of interest by using an antibody to that protein, and then on the RNA side, we're just going to use a piece of complementary sequence, an in situ hybridization probe that will bind directly to the target RNA. And then in either case, we'll attach on this digital spatial profiling barcode. So this is just an indexing oligo that allows us to know what target it was associated with. And we attach it using a photocleavable linker. And so now we'll be applying these onto just traditional slides. So these are just the same slides you would use for any other histology purpose. And you'll just affix tissue to that slide however you'd like. This gives you a lot of flexibility with how you want to interrogate your tissues. Obviously, if you have large tissue sections, you could just put one tissue per section. If there's smaller tissue sections, for example, corneal biopsies, or you're working in mice, or even if you want to create a tissue microarray, you can put as many samples as you'd like that will fit onto a slide. And then, as I alluded to before, our compatibility with the different fixation methods, whether you're using Formalin-Fixed, Paraffin-Embedded, fresh frozen, fixed frozen, all of these things are compatible with the platform. So it really doesn't matter about your sample type, about your sample fixation, or how you affix them to slides. Everything's going to be compatible here. And then your workflow is going to be really straightforward. If you're familiar with an IHC or an ISH protocol, that's exactly what we're going to do here. So we're just going to prepare your samples the same way you would for an IHC, just some deparaffinization and epitope retrieval. And then we're going to incubate with our reagents. So you're going to use some imaging reagents. These are going to be fluorescently labeled. So you can use fluorescently labeled antibodies, or we also have a collaboration with ACD. So you can use fluorescently labeled RNA-scope probes as well. These are going to be there just so you can visualize your tissue. So this is not what we're quantifying. So in addition, we're going to add these profiling reagents, those oligo-labeled reagents I showed you before. And so we'll combine them onto the slide so that not only do we have some morphology markers to be able to visualize our tissue, but also that really high-plex information sitting there invisibly on the tissue as well. So now we're ready for the digital spatial profiling barcode. And just as an asterisk here, I'll mention, while this is completely compatible with your manual tissue staining protocols, it also can be automated on a Lycobond system. Okay, so moving into our workflow. So again, we're just going to go through that process that I just mentioned, where we're going to take all those oligo-labeled reagents that have the photocleavable linkers on them. Again, these are going to sit invisibly on the tissue. We're going to combine those with some fluorescent markers, again, just for visualization, and that is what prepares the slide for the instrument. Then all we have to do is take that slide and bring it onto the GeoMx. The GeoMx has a slide scanner, so we'll be able to get an image of our tissue with those fluorescent morphology markers. And then we're just going to go through and select all the different regions that we want to interrogate. Here we're just looking at circles, but I'll show you on the very next slide. There's lots of different ways for you to interrogate your tissue and select the areas that you want to interrogate. Again, we have 4 channels, fluorescent channels to visualize our tissue, but our plex here is not dependent on those fluorescent channels. We actually can go all the way up to whole transcriptome on the RNA side, or about over 150, about 160 plex on the protein side. So really high plex information that you're able to get from all of these samples. Once you've selected all of these different regions, you can now walk away. The instrument's fully automated. So the instrument will go through, shine UV light at each region that you have indicated. That will then release the oligos into the buffer that it's sitting in. And then they're ready for collection. So there's also a liquid handling system that will come in, sip up those oligos, and deposit them into a well of a microtiter plate. So now we have all of those indexing oligos in a well of a dish, where every well corresponds to a different location of your tissue. And then the instrument will just repeat this for each area, each region of interest that you've indicated until it's collected every area. It could be a handful of areas, it could be hundreds of areas. It's really up to you and your experimental design. And then all you have to do now is take this plate out and read it out to get all of that high plex information. There is a readout on the nCounter for our low plex protein assays below 96 plex, but more likely you'll be reading this out on a sequencer. This allows you to get the highest plex on protein, or again, whole transcriptome on the RNA side. And as I promised, I'll show you all the different ways that you can interrogate your tissue. So, when we're talking about GeoMx, this is my favorite slide. I think this really highlights what GeoMx can do and how you can interrogate your tissues based on your experimental design. So as I mentioned, you'll have an image of your tissue where you get to select what morphology markers you want to see. So just depending on what you want to evaluate, you can decide, I want to look at tumor cells, I want to look at immune cells, glial cells, whatever your tissue is and whatever cells are important to you. And then based on that image, you'll be able to say, using geometric profiling, you'll be able to lay down some different shapes. You can use circles, squares, or there's even a polygon feature. So you'll be able to hand draw on that tissue and really define what areas you want to collect. This is, probably one of the most useful. Obviously, if you know your tissue, there's different tissue layers, or whatever the case may be, you'll be able to define that very clearly. And I'll show you some examples of this in just a few slides. We also have something called segmentation. So as I mentioned, we'll be using these fluorescent markers to define your tissue, but there's an algorithm built into the software where you can actually even use that that staining to define your collections as well. So in this case, we're looking at tumor in yellow and immune cells in this magenta color. And so you can actually come into this area and say, okay, I want to collect everything positive, yellow positive. and collect your tumor compartment, and even reverse that and collect everything else, or your tumor microenvironment. This will go down to a single cell resolution. So our third modality is what we call rare cell profiling, where even if your cells are interdispersed within the tissue, you can still collect them based on their staining. So here we're looking at CD3 positive T cells interdispersed within this tissue. So all we would have to do is use our geometric profiling to define an area. And then within that area, we could say, okay, collect everything red, in this case, T cells. We also have a contour profiling. This allows you to look at a proximal response. So you can draw tracks, as you see here. So here we're following a layer of invading immune cells. Or you could even, if it's kind of more central, you could draw some concentric circles around that area and see how a signal changes as you move distally. And then finally, if you want to ignore the morphology and just kind of sample across the entire tissue, we also have an option for a gridded approach. This grid is also user defined. So here we're looking at squares. You could change them to circles. You can designate how small or large they are, how close or far apart they are, and then just add it to your tissue in a drag and drop feature. So the main conclusion from here is all of this is user defined. You get to decide how you want to interrogate your tissues based on your experimental design. And I think the best GeoMx projects are the ones where people come in saying, this is what I want to determine, this is my experimental question, and then design their collection based on how to answer that experimental question. So I showed you some of the way that we are able to be very specific about the different cells and tissues that we collect. And the way that we're able to do that is with our optical technology. So within the instrument, we have 2 digital micromirror chips. Each one of those chips has 1,000,001 micron by 1 micron mirrors. And it's by turning them on and off that we fine tune that UV light signal to be very specific to the cells or tissues we want to collect. So here you can see these yellow cells that are tumor, in this case, within this area. This is the mask that the instrument will create, where everywhere in white is where the UV light will shine, and therefore that's what will be collected. And then again, as I mentioned, we can even reverse that and collect everything negative for that marker as well. So this is a really good example of how that was used. So this is a study, one of our earliest studies actually in 2020 from David Rim at Yale. And this actually ended up being a cover photo on clinical cancer research. But in this case, he was interested in looking at biomarkers within non-small cell lung cancer. But he had a hypothesis that maybe some of these biomarkers were only being expressed in certain cell types. And by collecting the entire area, he was kind of averaging out those signals and not able to really evaluate what those biomarkers, expression levels and how they change. So he came into his samples and decided to collect based on different cell types. So here we're looking at pan-cytokeratin to highlight the tumor, CD45 to highlight the immune cells, and CD68 to highlight the macrophages. And then the instrument will come in and then it will actually make a false mask so that you can actually see what will be collected in each region. So if you look here, everything in this light green color is collected in the tumor compartment. So that's one collection. Then the instrument will come back and do a second collection of everything in yellow, or the immune compartment. And then a third of everything in this light blue color, again, even though they're interdispersed, so collect all the macrophages in a third compartment. And then they did a fourth collection of this, what they call non-immune cell stroma, or basically everything else negative for the other markers. And so, just a quick conclusion from this paper, where they came into this work with only one biomarker, they actually were able to find 12 biomarkers that were cell-specific. And, so obviously all of this data is great, but if you can't get any insight from it, that is a limitation. And so part of what comes with the GeoMx is the data analysis solution. And so this is just a screen grab right from the instrument. This is exactly what it would look like. And so it integrates not only your images and the different areas that you collected, but all of the analysis tools and visualizations as well. So over here on the left, you can see, we'll be able to see the scan of your tissue and then every area that we collected from. And then here in the middle, we'll be able to set up all of our analyses. And a lot of this is done in a point and click manner. So you can see up here, we can just hit QC, scaling, normalization, whatever you need to do to be able to interrogate your data. And that will set up your analyses here. And then you can set up how you want to interrogate. So you can do it by different probes or genes. You can look at pathways. You can set up your own pathway groups or probe groups to interrogate. And then all of your visualizations will be shown here. Another thing that I think is really great about this software is you can also export at any point. So if you have some other analysis that you'd like to do, you can export at any point and take all of that data out to run whatever analyses you'd like. Or you can actually add to the platform as well. So there's a place for you to add custom scripts. So if you have your own R codes, or we also have a list of R codes that have been developed for the GeoMx. And I'll show you an example of one of them in one of our case studies. You can actually just plug it right into the software and start running those analyses as well. So you can always grow what you can do with this platform. So I'll start going through a few examples that I think really highlight some of the capability of the GeoMx. And so this was a study looking at the islets of Langerhans in the pancreas. And so in this case, they used some markers for the different cell types. So they used an insulin marker to highlight the beta cells, a glucagon marker to distinguish the alpha cells, pan-cytokeratin just for epithelium, and then of course a DNA marker. And so here you can see that we can kind of now visualize these different cells, but also different structures within these tissues as well. And then when we collect, we can actually collect based on those markers. So here we're only collecting alpha cells, and then here on the right, we're only collecting beta cells. And then we can start doing some of those comparisons. Here we're looking at a volcano plot of what genes are enriched in alpha cells versus beta cells. Of course, we get insulin in the beta cells, glucagon in the alpha cells. Not surprising, but of course, a good proof of concept. So just an example of how the morphology markers can be used to distinguish the different regions that you want to collect. So before I go into some more deep dives into some case studies, I do want to highlight some of the content. I alluded to it a little bit, but as I mentioned, we do have both protein and RNA content. On the protein side, we're at about 160 plex. We have it available for human or mouse. We have a fair amount of targets in immuno-oncology, also a lot in neuro, and all of this is modular and customizable, so you can decide which modules you'd like to run. You can do as many or as few as you'd like, and then there is room for custom antibodies as well. I'd say the biggest limitation to our protein assays is not plex, it's good antibodies. So obviously evaluating antibodies and making sure that they are specific to their targets and compatible with the platform are going to be the biggest hurdles. And so we've taken on all of that process. So every one of our antibodies modules have already been completely validated and optimized so that you don't have to do any of that work. Everything is ready to go out of the box. And then on the custom side, the biggest hurdle is being compatible with IHC. So if you've done some IHC with some antibodies and they're really important to your research project, you can just get them tagged and added in as well. And then we also have a collaboration with Abcam where their antibodies are available to be ordered already tagged for the GeoMx as well. On the RNA side, we have no limitations. We go all the way up to whole transcriptome. So we're looking at over 18,000 genes in the human panel, over 22,000 genes in the mouse panel, every protein coding gene within the transcriptome. So you really get true discovery spatially with this platform. All right, so I'll start going through some case studies at this point. Just picked a couple of my favorites that I think kind of highlight how the GeoMx can be used and what the capabilities are. This is a study out of the Broad Institute, out of the lab of Jonathan Chen, where he was really interested in looking at microsatellite stable and unstable colorectal cancer. So in this case, he was not only looking at the primary tumor in the colorectal cancer samples, but also was able to get some tissue from lymph node metastases so that he could also evaluate the metastases as well. So what I'll orient you here first. First here we have the primary colorectal tumor over here on the left. And then here we have the lymph node metastasis tissue on the right. And then for each one of these circles, we have a blow up here. So just to give you an idea, what we're looking at here is just the tissue evaluated within each one of these circles. And then in green, we have a pancytokeratin positive tumor. And then the magenta is immune cells. And so what we are looking at around these circles, each one of these bars indicates expression of a gene within this area. And in green, we're looking at the tumor. And in this light blue color, we're looking at that same expression of the same gene within the microenvironment. And so what they really highlighted, or what they focused on, was this IDO1 cluster. And so what they were hoping to determine is if the areas of metastasis were originating, which part of the tumor they are originating from. So they collected areas from the margin, areas of low-grade tumor, also areas of high-grade tumor in the center, and then compared that to collections in the metastasis. If we focus in on this, on the pathology or the morphology of this tissue, the hypothesis going into this study was that the metastasis originated from the areas of low-grade tumor. The tissue looked much more similar than when you compare it to the areas of high-grade tumor. But what they were able to find by using GeoMx and the whole transcriptome is that the IDO1 cluster from the areas of the high-grade tumor was almost identical to the IDO1 cluster in the metastasis, suggesting that these metastases actually originated from the areas of high-grade tumor. I'm going to switch over to another case study. This is a case study out of UC Riverside, out of Byron Ford's lab. He actually just recently moved to Howard University as an associate dean. But this is a really interesting study because I think it really kind of highlights why spatial is important. So just to give you a little bit of background, Dr. Ford studies ischemic stroke, and he uses a mouse brain model for his studies. And what he would typically do is just use RNA-seq to try to determine differential expression in the areas of damage versus areas that haven't been damaged. And so, obviously that produced lots of data, but really wanted to know how spatial information could supplement these studies as well. So this is what these mouse brains look like. So here, this dark region here is the area of damage or the ischemic stroke. In addition to looking at different time points, so 3 days post-ischemia and 7 days post-ischemia, he also wanted to look at the spatial context of these different tissues. So he collected areas of the tissue that are close to the area of damage, some that were further away, and then some on the opposite hemisphere. And so once we interrogate that data, we learn a couple different things. So the first thing he wanted to do is he wanted to see how different the spatial information would be from his traditional method. So he compared his traditional method where he would have taken all the areas on the ipsilateral hemisphere, or the one with the damage, and he compared it to all of the expression on the contralateral hemisphere, or the opposite hemisphere. Here you can see we get a lot of data, about 300 significantly changed genes when we do it this way, and kind of ignore that spatial context. But if we just focus in on the spatial question, if we just look at the areas of the brain that are very close to the damage as compared to the areas of the brain on the opposite hemisphere, now we actually see 7,000 genes differentially expressed. So just a huge amount of data that can be lost if you aren't taking into account that spatial context. And then again, here we can see within that region where the damage, where that damage is, that what we're calling the core border, you can see there's just a significant amount of differential expression, even compared to the regions right next to it. And then one of the different analyses that you can plug into the software, so this is one of those R codes or supplemental scripts that you can add in, is a cell deconvolution algorithm that allows you to use single cell RNA-seq data and different phenotypes of different cell types to plug it into an algorithm to deconvolute lots of different cell types. So in addition to all that differential expression data, he can also take a look at all of the different cell types across across his tissue and in the different areas of collection. So, obviously a lot of these conclusions we just went through, just a huge amount of data that he was able to get from this spatial question. But he also just didn't stop here, right? As I mentioned, that GeoMx is a multi-analyte platform, so we don't have to stop with RNA. So he also wanted to look at the protein as well. So same sort of design, same sort of collection strategy where he collected near the regions of damage, a little bit further away, and then on the contralateral hemisphere as well. And then if we look at the comparison here as well, we can now see that if you look at one hemisphere versus the other, we get about 4 significantly changed proteins when we take into account that spatial context and only look at the areas near the damage versus far away. We now get 27 significantly changed proteins. So again, just a lot more data we're able to get by including that spatial context. And then again, we could use that proteomic information to then also look at profiles of the different areas as well. Again, I won't read the results to you. I'm happy to pass along these slides, but again, just a lot of different information that you're able to get with this high-plex information as well as that spatial context. And then this is a study that I mentioned earlier. In that I just wanted to show you how valuable geometric profiling can be. And so this is a study looking at embryogenesis in the mouse. In this case, there's not going to be markers for every different tissue layer and organ that you're interested in. So, but because embryogenesis is so widely studied and known, we actually know a lot just by being able to look at these different tissues. So if we look at these different tissue time points, we can actually, just based on some simple staining, so here we're just using a DNA stain, an epithelial stain, and some neuron stain, we know enough about these layers that we can actually just use our hand drawing feature to really distinguish all of these different layers and collect them all separately so that we can, in this case, follow gut development from embryonic day 9 to 15. And you can see how much more developed it gets here. But even with these really defined structures, because we actually can just hand draw, we can be really specific and collect those different layers separately. We can do the same thing in the lung here, starting with some very basic structures. As they get more developed, we can still follow them along and only collect the different areas that we would like to collect. And then again, all the way across an embryo, we can collect all of the different tissues. So they're all listed here. So we can follow that embryogenesis through these different samples as well. So here you can see here, we're looking at a limb, bud, brain. And again, we don't need markers for each of these. We can just collect them based on what we know about embryogenesis. And what's great about this is in one study, they were able to find different cell types, embryonic cell types that they hadn't been able to find even using single cell RNA-seq. So this is a study that came out kind of trying to identify the same thing in 2019, but they were able to find a ton more cell types and different information by being able to really focus in on the different biology that they were interested in. So the last case study that I'll take you through, this is a group out of Mass General Hospital. I see this is kind of hard to see, but this one I really like because it highlights a couple of different real advantages of the GeoMx. And in this case, this was a group that got some of the first COVID samples of the pandemic. So they got these samples March, April of 2020. These samples were really precious. They were, trying to be distributed to lots of different places. So they only had a finite amount of tissue to try to get as much information as possible. Again, since these were COVID-infected tissues and also early in the pandemic, they weren't taking any chances. These samples were fixed in formalin for 21 days. So we're talking about really fragmented RNA, really poor quality samples, but obviously really important to get as much information as possible. They had 5 different patient samples of autopsy samples, and they wanted to, again, try to get as much information as possible. And they told us that's why they came to GeoMx. So they knew that we would have the best compatibility with FFPE samples, but also the GeoMx is multi-analyte, so they could evaluate both protein and RNA on these samples. So this is what their design looked like. They wanted to evaluate a few different things. They wanted to know where the virus was in the lung. So they used an ISH probe to SARS-CoV-2 so they could visualize where the viral load was. And then they also used some morphology markers, some fluorescently labeled antibodies, pancytokeratin for epithelium, CD45 for immune cells, and CD68 for macrophages. So in addition to that, they'd have some information about the immune response. So they ran an RNA assay. This is back before we had whole transcriptome. So we had something we called the Cancer Transcriptome Atlas, which was really immune and oncology focused. It's about 1,800 genes. But even from these 1,800 genes, they were able to then incorporate their single-cell RNA-seq data and then visualize. Here we're looking at a dot plot where we're visualizing all of these different immune cells across the tissue based on where the virus was located. So red being a SARS-CoV-2 positive area and blue being a SARS-CoV-2 negative area. And so now we can get a sense for that immune heterogeneity within that tissue. But then on top of that, of course, they also get their High-Plex information, tons of differential gene expression that they can evaluate based on viral load. They could also look at a t-SNE plot to see how their different regions cluster. Something I thought was interesting is all of their samples clustered based on the patient rather than based on the viral status. And then, of course, also lots of pathway analysis and High-Plex information that can be gathered. And then, of course, as I mentioned, it's multi-analyte, so they're also able to get protein information as well. So here we're looking at protein data across these different samples. So, again, just highlighting some of the different statements that they mentioned in their paper, of course, that, some of these insights wouldn't have been able to be gathered any other way. And, they do make mention of the fact that they didn't expect to be able to evaluate RNA in these samples due to the really high fixation of these samples, but the GeoMx, based on the chemistry, just works really well with those sample types. And this is just a publication that came out of our biostats group, actually. And this is the algorithm that allows you to do that deconvolution. So here we're looking at a lung tumor. We do have a deconvolution algorithm for lots of different tissue types, however. But in this case, we just gridded the entire tumor. And so I want to just highlight one thing here. As I mentioned, if you don't want to pay attention to the morphology, you just want to grid across, you can do that. And you would just do it in a drag and drop fashion. So it would just add the grid to the entire tissue. The nice thing here is then we can go through and we can delete any of the squares that we don't want to collect, right? If we don't want to spend the money or time sequencing them, we can just delete them. If they land on a blank space, we can just get rid of it. And then in addition to that, in each one of these squares, we're actually doing 2 collections. So we're collecting the tumor in green and the microenvironment in red. So we actually are able to not be limited by that square or circle. We can just separate them out into their tissue types within those areas as well. And then from that information, in this case, we're deconvoluting immune cell types. Here we're looking at the 18 different immune cell types we're able to deconvolute in this case. And then across this tissue, in each one of those squares, we can now take a look at the abundance of each one of those cell types. It's a little hard to see here. There's some pie graphs that correspond to this pie graph. I think it's a little bit easier here with the dot plot, where now we can actually visualize that immune heterogeneity and the landscape of immune cells. So we can see, maybe monocytes are clustering all over this tumor, but then CD8 naive T cells are only clustering here on this left side. So just a ton of information we can get from this, this platform. And then I mentioned that we do have a lot of publications, about 5,200 across all of our platforms. But I think we actually just passed 160 peer-reviewed publications to date, just using the GeoMx. Tons of really high impact journals that they were published in, as well as some cover photos. So this is the one that I mentioned. That's a cover photo from that non-small cell lung cancer study. Here's another one. We actually just got our third. So in addition to just really insightful data, also some really pretty pictures that are definitely being recognized. But again, all of these publications are really interesting, kind of showing how GeoMx can be used, and they're all available on our website if you kind of want to browse around and see what's being done. The other thing I'll mention is, the time to publication has been really kind of impressive too, right? So the GeoMx has only been on the market about 3 years. You can see by the number of publications, but just for an example, that COVID study where they got their samples in March and April of 2020, that paper was published by either November or December of 2020. So just a really fast, really quickly how much data and what high quality data you're able to get. And I highlighted, I think, all of these collaborations throughout the talk, but I'll just put them all in one place. As I mentioned, we have a collaboration with Leica, so the entire workflow can be fully automated on a Leica bond system. Of course, manual works just as well, so you still do have that manual workflow. We have a collaboration with ACD, so we do have compatibility with their RNA scope probes to use those as morphology markers as well. And then a collaboration with Abcam, where every one of their antibodies in their catalog are also available to be tagged and added to the GeoMx as well. And so I'll just kind of summarize what we've talked about so far. As I mentioned, really high plex here, and we haven't reached a plex limit at this point. So you actually can add custom to either our RNA or our protein. We're already over 22,000 on the RNA side, but if you wanted to add any additional genes, maybe transgenes, non-coding genes, whatever the case may be, there's still room to add custom there. And then on the protein side, as I mentioned, we have over 150 validated antibodies, but also room for custom on that side as well. This is a really high throughput platform. This instrument holds 4 slides at a time, so you can get through 4 slides in a couple hours on the instrument. So just a ton of information, up to 60 samples per week at any plex level. We talked a little bit about the sample compatibility. So again, FFPE, fresh frozen, fixed frozen, whatever the case may be, all perfectly fine, as well as whole mount or tissue microarrays. We touched on it being multi-omic, both RNA and protein, and then fully automated as well. And so I'm going to take the next couple minutes to just kind of highlight what's coming next. As I mentioned, we have adapted our initial chemistry to really bring us into this spatial market. And so where the GeoMx is that really high plex platform and really high throughput, looking at tissue and cell types, we've also been able to adapt this chemistry to get us to an imager as well. So we have the Cosmic Spatial Molecular Imager launching this year, so in the next couple of weeks or a month or so. And so this allows you to get a really high plex down to 50 nanometer resolution of transcripts or protein within your cells. The compromise here is going to be plex and throughput. Obviously, it's an imager. It's not going to be as high throughput. And right now we're at about 1,000 plex, but that is increasing as well. So now we're getting down to our single cell and subcellular resolution. And so much like GeoMx, we do have a large panel. It's not whole transcriptome, but it is 1,000 plex and increasing, has that same compatibility with sample types. It's also multi-omic. Here, instead of 60 samples a week, we're looking at between 2 and 20. Again, imagers are going to be much slower, but that much higher resolution. And so just to give you an idea, this is a melanoma study where in one run on the instrument, they were able to interrogate 4 million cells and 2 billion transcripts. So this is about 15% of an entire cell mapping, cell atlasing project that used 406 different labs. Again, this is just kind of highlighting what's coming next. In addition to GeoMx, we're not getting out of spatial biology anytime soon. So lots of new things coming on the spatial side. So I will just kind of show you, this is just a few different samples that we've run, again, from FFPE to fresh frozen. What we're looking at here is different cell typing within these samples. So just a ton of information you can gather here as well. So that will do it for me. I'll stop here and see if there are any questions. Thank you so much for having me.
Thanks for coming. This is a great presentation.
Thanks,
Yeah,
Alex.
so does anybody have any questions? Because if not, like Pat said, I do have a couple questions of myself if nobody else has any. Okay, well, I'll start with one then while everyone else is thinking. So kind of as you were talking about, it sounds like really the design is really a critical part to planning these experiments. And I was just wondering if after you've done like let's say you do the work with the initial slide that you're working with, can the slides be refreshed in a way? Like, can you add a new, the cleavable portions? Like, let's say you do an initial interrogation and you find something interesting, and you say, "Oh, now let's go back and we want to, maybe select some slightly different groupings or subset these cells a little bit differently." Is that something that's possible?
Yeah, you can. So it's a little bit harder on the RNA side. Obviously, once we do that epitope retrieval on the RNA side, the RNA is being exposed to elements. So your time limit is much shorter than on the protein side where, we can strip off antibodies and reprobe pretty easily. But we're also just using 5-micron sections. So, if it has been longer, a lot of times you can just use a serial section and get that information as well. In some cases, if you don't really have your experimental question fully mapped out, the gridding approach can be one way to go. You have all that data, you can always come back to it. But I feel like that usually lends itself to a second project anyway, because now you're like, well, I really want to know about this cell type or tissue type, which you've kind of ignored in that first project. So it really just depends.
Okay. And then another one I've got while everyone's still thinking. So it sounds like it kind of was structured, but so since you would need to get kind of multiple slides in some cases to really have the replicates to do some of these more statistical tests, it sounds like you could define your regions, whether it is based on specific probes, or I guess if you're doing the grid selection that way, you can base it off the first slide and then use that for, let's say you're doing another 20 slides, you could not have to manually go in and reselect those. It'll just kind of automatically select everything.
Yeah, absolutely. You could definitely do that. And then the statistics built into the software account for all of that as well. So not only multiple samples, but multiple testing per sample. So all of that's kind of built in. But yeah, the actual ROI collection. So if you did it on one, you could reapply it somewhere else. But of course, if you're doing multiple different samples, but maybe all, colorectal cancer or whatever the case may be, you may have to drop your circles or whatever down yourself, but then the actual algorithm that separates it out into their tissue specific collections is automated through the software.
Okay, cool. Thanks.
Of course.
We do have a question that came up. So it's a question asking if this is a service that Nanostring provides, or if we would have to buy the equipment and reagents. So like if in this case, if the investigator has fixed samples, can he send them to us for analysis? Or I guess, the question being, does Rutgers already have this equipment in place too?
Yeah. So it's a unique time to answer that question. So I'll answer on the Nanostring side. So we are not built to be a service provider. So it is typically a model of where groups would buy an instrument, and then obviously we help support that. There are groups that will take outside projects, and we're happy to suggest that. And then we are, hopefully Rutgers will have an instrument soon. There are some conversations ongoing.
Yeah, and I'll add to that. There are definitely conversations about putting a couple of these instruments in different locations at Rutgers. If it's something you are really interested in, please reach out to me and Alex so we can let the people who make these ultimate decisions know that there's a lot of interest, and that might help speed up the acquisition.
All right. Any additional questions from anybody? No? Okay. Well, then I'll say thank you for an amazing presentation with all the fantastic information and the pictures that we do have one more question. So what about the analysis and training for the analysis?
Yeah, so I actually, I always say I'm always surprised how integrated Nanostring is in that. So included in the instrument would be the entire training process as well as data analysis training. Rutgers does have their own application scientists that will help with data analysis support. So all of that, the software is actually relatively user friendly. It's a very point and click software, as you could probably see from my screen grab. But we do have a lot of resources to make sure you have everything you need there. And then in addition to that, we also have something called our spatial data analysis service, where if you don't want to be bothered and run your own, we also have a team that will do data analysis for you for a fee as well. So across the gamut, we'll make sure that you're able to gain insight from your samples.
Perfect. Cool.
Okay.
All right. Well, then just finish it up. Yeah, thank you very much. It's fantastic. And yeah, if anybody has any questions, like Pat said, please reach out to either myself, Pat, or the Nanostring team, and we'll work on getting some of these systems installed. Or if you have any questions just about the general functioning, and we can go from there. So thank you very much for joining today, and look forward to seeing everyone at the next seminar.
Thanks,
Thank you,
everyone.
everyone.
Thanks for having us.